Hello, and welcome to everybody to the fourth installment of the fall 2021 Sara Turnbull Visiting Designer Speaker Series, where we talk to designers, artists, activists, and in this particular case, researchers, scientists, to create a broader conversation about design here at Lehman. My name, again, is David Schwiddick. I'm an assistant professor of graphic design and digital media in the arts department here at Lehman. And today we're going to be continuing our discussion about the eye. And this is all in conjunction with the Eye's Habit. It's an exhibition that's in the art gallery at Lehman College. I suggest you go see it. I happened to just pop in yesterday. It's amazing, but I hope you can also pop in and see it. But today we're going to be totally shifting gears in some ways. And we're actually going to be literally talking about the eye, the human eye, and just eyes in general in the animal kingdom as biological structures, right? Our guest today is Professor Stephen Redenti. He's an associate professor of biology and biochemistry here at Lehman. He's currently heading up the Redenti lab, which is tasked with exploring cellular communication. It's involved in the development and regeneration of retinal and other central nervous system tissues, which he's going to be talking about today. His lab is particularly interested in how cells respond to morphogenetic fields of discrete, localized biochemical and electrical signals, leading to the development of organized neural tissues. I vaguely understand what that is, but he is also going to explain. He's going to explain that as well, I hope. Professor Redenti's research is focusing more broadly on investigations of migration, cell-to-cell signaling, and regulation of retinal development, the design of man-made structures that mimic biological structures in order to enhance cell-tissue interactions. And also a branch of this work has also led to the analysis of drug delivery and drug polymer encapsulation toward neuroprotective and anti-cancer applications. So first thing, this is an art and design lecture. So you may be wondering what the heck Professor Redenti is here doing. He's a scientist. He works with data and in a lab. But actually, if you could you actually have a background in the arts. Could you just quickly tell us about that, Professor Redenti?
Yeah. Oh, thanks for having me, David.
Of course.
I've been wanting to return to art for years, and
you're welcome anytime.
When when I was an undergraduate, I had a degree in sculpture and biopsychology at the same time. And then I worked in sculpture for a few years, apprenticed with a couple of artists, and moved to Chicago, worked with some artists there. And then I applied for a master's in fine art and a PhD in biology, and I was accepted to the PhD program in biology. But both were very fascinating for me. So in some ways, I consider my work as a scientist as an extended MFA project.
Interesting. So you are going to talk to us today about your research, right? And you have a presentation to show us. Why don't you show us what you've been working on?
Sure. And I'm going to share my screen. I can't see anybody in the audience. I guess that's
no. That's one of the downsides of these webinars. That
it is. Okay.
It's like a real seminar where the lights are down and it's just you on a stage and you can't really see anyone. Think of it that way.
Yeah, that's good. All right. So anyone out in the audience, if you want to talk to me about art or collaboration or science, anytime, my email address is, just contact me or David anytime.
Yeah, of course.
And I would love to work with artists again. So now I'm sharing my screen. You see that, David?
Yeah, I could see it just fine.
Okay. So we can have this as a kind of a conversation or
sure.
a lecture based on principles of biology that drive the evolution, architecture, and bioengineering of the retina, and try to present it in terms that are understandable. But I do want to give you the context of evolutionary forces driving the design of the eye.
Right.
And we're going to talk about it from the simplest forms, from single-cell organisms to the human retina and eye, right? Which is what I'm working on presently.
Right.
And so I give you this. Should I just go right into it, David?
Yeah, just launch right into it.
Okay, I shall launch right into it. So I put a quote down here from Charles Darwin, and there are some selective forces you want to keep in mind. You may be studying engineering or design as art students, but the material that's being sculpted or shaped in evolution is life itself, cellular life in particular. And the forces that are used to manipulate that matter would be selective forces, forces that are driving survival of a species. So this is happening continually over time, over great expanses of time. That's how you get variation and diversity in biologic forms. And Charles Darwin said it's not the strongest of the species nor the most intelligent that survives. It's the one that's most adaptable to change. If you keep this in mind as you think about evolution, some of the simplest organisms like prokaryotes or simple bacteria are the most diverse and longest living on the face of the earth. And so that's, with that, I'll move into the origin of cellular life to begin. And most slides will have a short video. And if you take a look down at the right, you'll see a computational model, probably using a software called Maya, if anyone's interested in biologic modeling, to simulate the origin of the first cell at the bottom of a volcanic well, a hydrothermal vent, where fatty acids are pooling together and self-assembling into a lipid bilayer, a fatty bilayer that will form the first protocell in the early oceans. And this is predicted to have happened about 3.5 billion years ago. But this animation, I think, will help you visualize it. It's not just that they formed at the bottom of these boiling vents, but then the water erupted. The video will show you this. And then it evaporated on the muddy shores. And as it evaporated, the fatty acids got pushed along to the edge and an aqueous center was preserved. And that's science's most current explanation for how the first protocells evolved. And you can see that happening down at the bottom right.
That's the basis of any cell membrane, right? A lipid layer surrounding an internal structure, right?
That's it. It's very
basic.
Universal. Yeah. It traps the ocean within us, so we could, survive. So, and I put a geographic timeline, a geologic timeline here at the top, just to give artists and scientists in the audience an idea of, the timeline we're talking about. We're going from the origin of the Earth 4.6 billion years ago to here we're talking about the first cells about 3.5 billion years ago. And then we come to the first cells with a nucleus, which I'm going to go to on the next slide. These are eukaryotic cells. That means they have just DNA enclosed in a nuclear envelope. That's you and I. We are eukaryotes. And, E. coli would be a prokaryote, the bacterias. And we're going to start with these. And if you want an idea of how briefly the human species has been on Earth, you can take a look here. It's about 200,000 years that our species has existed on planet Earth. A recently, newly designed biologic form, doing good and bad for the environment. So I just want to convey that to you. Those are the building blocks of life, and we're going to take it from there. And I start all my introductory bio courses like that. And so we move on then to the first eukaryote, single-celled creatures swimming in the early oceans with eyes, not the type of complex eyes that you and I have, but simple eyes. That one term I'd like you to sort of grab right away as artists is acceptation. And this reminds me of found object art that, maybe I studied when I was younger, where you just take disparate pieces and put them together and form new objects. So acceptation is an example of how the early eyes formed in these three single-celled eukaryotes in the beginning. Maybe 200 million years ago, there's a dinoflagellate, and these dinoflagellates are quite concentrated in the ocean waters, but they're a simple organism, and their eye looked like this. If you take a look to the left here, you have a repurposing of mitochondria to form a layer of the eye, a lens of the eye, and this sort of retinal body, which is the photoresponsive part of the eye. That wasn't the original intent of nature, but through trial and error or through the time passing, this small eye-like structure formed. And it looks like a complex eye, but it's far from a complex eye. It allowed the animal to navigate away from or toward light. If you wanted to know what it was like to see the world through a dinoflagellate's eye, you could probably close your eyes and navigate with your eyelids closed. And that's probably the amount of resolution they had. And why would you want to move toward and away from light? To feed and to protect yourself from being, eaten. And we move up to these other chlamydomonas. This is a rhodopsin molecule, another repurposing of molecules existed inside the cell. And then we can move over to blastocladelia. And these again are lipid-filled vesicles with mitochondria being repurposed. And so if you follow the timeline here, you have 200 million years ago, these creatures are emerging. About 1,000 million years ago, this creature is emerging. And about 400 billion years ago, this creature is emerging. Not at different times, these different life forms are co-opting their built-in components to give themselves some form of vision. So you'll see this.
That still isn't very long ago. I mean, I know it sounds like you're talking like hundreds of millions of years, sounds like a long time, but in the history of the Earth, that's really like an eighth of our history as a planet, right? It's not a long time.
Yeah.
In the grand scheme of things. Okay.
It's a way to look at it. Yeah.
Yeah.
Sometimes adaptation happens quickly. Sometimes it takes more than 100 million years.
Right.
These are just a couple of examples. I forgot to point out, but this is a tiny dinoflagellate swimming in the top right-hand corner for you to see how simple this creature is. And you can even see this, the eye is called an oceloid. It's this little dark pigmented structure in the middle of its body. So that's the first sort of eyes in eukaryotes. And then we jump forward to some structures that might look more familiar to everyone. And I give you the million of year ago time for each. MYA means the simplest eyes that start to resemble a complex eye are in these little structures called lipids. These little creatures. If you've been to the beach, you've seen barnacles on the sides of rocks, and always populated and interspersed between the barnacles are these small shell-like structures but they're flat up against the rocks. Those are lipids. They're an ancient species, and they have a snail-like body underneath their hard shell. So their photoreceptor layer is like this. It's just a flat sheet of photoreceptors with nerve fibers. And this allows them to do phototaxis, which means they can be attracted to light. And how did these emerge? Over millions of years, their skin cells acquired the properties of photosensitive cells. That would mean that if light hit these cells, they would depolarize or have an electrical potential that could be transmitted down the nerve fibers. Then we move up to this. This is a shell mollusk, and this is the first time in evolutionary history you'll see the invagination of a cup-like structure in the eye, and that cup-like structure is going to be important. You'll see in the next couple of, when we get to the octopus and the human eye. But these are slightly more complex, and this would provide more complex detail about the visual world around them. And so they have pigmented cells, and they also have photoreceptor cells. And pigmented cells are useful in the evolution of eyes because they absorb light. They prevent the scattering of light. So you could start to imagine that the world of the shell mollusk would be slightly higher resolution than the lipid, and that would provide several advantages, survival advantages in the Darwinian world for survival and feeding and reproduction. Then we move forward to the Nautilus. You probably are familiar with this creature. It's got a beautiful spiral shell, and it looks like it has octopus tentacles pointing out of it, and it swims backwards, has a big, beautiful eye. And this is the first creature in evolutionary, in this example, where they have a pinhole. And the pinhole gives you some fixed distance between the incoming light and the retinal sheet. And that gives you directionality. Rather than just sensing the presence or absence of light, now you can sense the direction from which the light is coming. And it gives you a little more intelligence when navigating a light-filled environment or dark environment. I'm moving steadily along here, but then, so we have 135 million years ago, the octopus emerges in the evolutionary timeline and has a complex eye. It looks like a human eye on first glance. I've been looking at eyes for 20 years now. I can't believe that, but it's true. And what you have then is a lens for the first time, and then you have a complex retina. A complex retina, but it only has one type of photoreceptor. You know, the human retina, I'll tell you in a few minutes, has 4 different photoreceptors. But this has a lens, which increases the resolution. It also has a cornea, which protects the ocular organ. These are just skin cells or water. And then it has an optic nerve, sort of the emergence of a proper optic nerve. And so one question that if you ever looked at an octopus eye, I was going to post an image of it here, but if after the seminar, if you'd like to Google octopus eye, and you'll see it has a ruffled eyelid. And the ruffled eyelid allows it to control the colored wavelengths that are coming into its eye, so it can have a primitive form of color vision with a single photoreceptor. Pretty ingenious. And then the subtleties of evolution, they're hard to explain, but that you wind up with these brilliant structures that engineers would have a hard time competing with. Still do, especially when it comes to the eye. Should I go on, David?
Yeah. Well, I do have a question, but we can wait until the end, which is to say, if you had to design an eye, a human eye, would it look like the one we see here? But I suppose you could probably answer that after you go on a little bit, because you might have some insight into that in your slides.
Yeah, we'll talk about that. That's a good question. Yeah. I can think of one very important change I would make.
Right.
So, namely that the nerve tissue could regenerate, but that might have its own problems. But so this is the modern primate eye. It's not, the human eye obviously didn't emerge 44 million years ago, but that's when the last branch of primates, New World primates, emerged from Old World primates, and that's when the sort of modern vertebrate eye, mammalian eye that you see here emerged. And so let me point out the structures to you as design students. So in case you wanted to build an eye later today, a modern mammalian eye, it'd probably be easier to start with the limpet, but you have clearly, as many of you know, as college students, you have the cornea, and then you have the lens. And this is unique. It's different. An octopus lens cannot bend. It doesn't bend or adjust to accommodate the field of vision, but the human eye can bend and accommodate vision. It's sort of a flexible, it would be like having a flexible lens on your iPhone, although your iPhone has so much intelligence now, it's probably not.
Yeah, it doesn't need it.
It doesn't need a flexible lens. Yeah. The iPhone 11 has 3 lenses. It's amazing.
Yeah.
Super eye.
It's all done by software. That's the thing that's another interesting point that I wanted to talk about, but continue though.
Okay, so let me get to the back of the eye. We move through the vitreous humor, which helps to contain the incoming signal. It's basically water. And then you get to the retina, where we're going to spend the rest of the talk, just a few slides on its structure and how scientists are trying to engineer them. But the fovea is where's my little indicator? The fovea here, I'd like to draw your attention to, and I'll talk more about it in a second. This is the point where you and I will see detailed features on the faces of people we know. This is how we can distinguish friends, and this is how we can look at a slide like this and read the text and see the different colors and the different structures. This is where the cones are concentrated. It's not present in mice, for example, but it's present in primates. This fovea gives us really exquisite, detailed vision. And then the rest of the retina is spread out. Let me see if I can get it spreads out to the edge of the eye. And there's the optic disc. The optic disc is where the axons from the retina exit the back of the eye and go to the visual cortex in the brain. And this is enough for a comparison. I wanted to essentially compare the limpet to the later evolved eyes, and then compare the octopus eye to the human eye to show you some of the differences. Mainly that we have a flexible lens, the octopus eye does not. We have 4 photoreceptors that allow us trichromatic color perception. The octopus does not, but it has a ruffled eyelid. And that's that for that slide. And so I'm going to state here, natural selection favored gene combinations that increase the functional efficiency of the eye. So you could be sure that many members of each species died in the process over millions of years before the eyes we're looking at today emerged. It's trial and error, and it's survival of the most adapted, as I mentioned on the first slide. And that's this extensive geologic time, trial and error, massive losses, and the emergence of a sort of optimized camera-like eye This is how you can account for the complexity of the eye, time and trial and error. And now I bring you to this, where I've been in this for 20 years, looking at the structure of the retina. I started when I was a doctoral student at Hunter College, working on the biochemistry of these structures. These are the photoreceptors and the first synapses. So I'll show you this. I have a little video here. Let me show you how we can break the eye apart into its different components. You have the conjunctiva on the outside. It's a sort of a mucus-like film. Then you have the sclera. The sclera is the tough, leathery substance surrounding the eye. You have the choroid. The retina has a lot of oxygen needs and a lot of need to remove metabolic waste. It's highly vascularized, and this red cup shows you how much there is a huge vascular bed underneath the eye that provides nutrients to the photoreceptors. And then you have the retina itself, which is the orange cup. And then we move closer inside to the vitreous. So I'm going to move forward now into the retina's architecture and function. And the distance between the outside of the cornea and the retina is about 22 millimeters. It is 22 millimeters. And there's pigment epithelial cells. Another difference between our eyes and octopus eyes is that our photoreceptors face backwards. The photoreceptors that detect light and detail for us are actually poking backwards into the pigment cells in the back of our eye. And that's a way to clean up the noise. We've evolved this for greater detail perception. Octopuses, their retinas face outward. Their photoreceptors are pointing right to the outside world. So they would have a noisier incoming signal. Okay, so the pigment epithelial cells are the most active. They call it phagocytosing cells in the body. They're the most active garbage consuming cells in the body. This is one of the highest metabolically active sites in the body, because every time a rhodopsin or an opsin molecule is used, it's spent and it has to be recycled through the retinal pigment epithelial cells. And rhodopsin and opsin, these are the photosensitive proteins inside the photoreceptors that allow light to be detected and then an action potential to be generated. So if I walk through the architecture of this creature, we have the retinal pigment epithelial cells, and now I'm just going to drop down the left column for all those who have not seen this and diagrammed it for years and years. You have a photoreceptor cell layer. You have the outer limiting membrane. That's really just the membrane between the inner retina and the outer retina. It's where the outer segments of the photoreceptor are separated from the rest of the retina so light can hit it properly. And then you have the outer nuclear layer. These are just the cell bodies of the photoreceptors. And I chose this diagram. There are thousands of diagrams of the retina, but I thought it was artistic. I think it has a good aesthetic quality. I'll have to ask the designers in the audience.
Oh, it's beautiful. It is.
Excellent, yes.
Yeah.
So the outer plexiform layer, I probably spend too much time looking at the aesthetics of scientific images coming from an art.
That's okay. That's okay. We like that.
Then this outer plexiform layer, this is the first synaptic connection between your photoreceptors and the inner workings of your visual system. And then you move to the inner nuclear layer. These are the second order cells, the second neurons in the visual pathway. You have the inner plexiform layer, a massive synaptic layer. And then you have ganglion cell layer. And the ganglion cell axons form the optic nerve that go through the back of the eye and synapse in the visual cortex. But one thing I want to tell you as designers is that there's a high level of convergence at the retina. If you were to build an instrument like this, there are 100 million, approximately 150 million or 157 million photoreceptors. And there's about 1 million ganglion cells. So even at the level of the retina, you've got 157 million cells detecting in the human retina. And then a lot of processing and information being consolidated even before it reaches the ganglion cell. This is sustained through the optic nerve to the visual cortex, where it's kind of reanalyzed and parsed out into its different components. So that's the retina, and those are the structures I'd like to point out to you today. And there's I didn't mention the cell types, but there's horizontal processing that occurs. Here you can see H, these are horizontal cells. So it's not just one rod cell to one bipolar cell. The bipolar cells are in the middle, but approximately 11 rod cells will synapse onto one bipolar cell. So they become fields. Each pool of photoreceptors becomes a visual field in itself. And if one population of rods or cones are active, it's set up in such a way that the neighboring neurons will be inhibited to turn down the noise, and the gain will be turned up on the neurons that are being stimulated. So a lot of intelligence built into the architecture and the functioning of the retina at this level.
Yeah, I mean, it's hard for like a lay person to look at something like this and try to fathom, even through like a process like evolution, but by way of natural selection, how something like this could be reduced to something very basic, like what you started with, right? You were starting with these very primitive structures, and I kind of wanted to talk about that a little bit. So we're talking about, as long as we're like talking about art and design, right? Since we're in the context of art and design here, I wanted to talk a little bit about intelligent design, which is a really loaded term for, and it has been for, well, since Darwin, right? But generally speaking, intelligent design in a very general, non-loaded way kind of describes what designers do, right? Like human designers, me, our students, etc. They organize and create with the intention of achieving certain objectives, right? In this case, the objective would be to decode visual information into something the visual cortex can understand and work with. But when people say intelligent design outside of the sphere that we're talking about, they mean it's an alternative to evolution by natural selection, right? That there are certain irreducible complex structures in biology that really can't be explained any other way. So holds the belief, right? And it is a belief. It's not a theory. It's not testable. It doesn't have any predictions or anything like that can be tested. So the human eye is, as you've just explained, is really complex, and it's really difficult to see how it could have evolved on its own. But your research suggests, and what you've said today, is that it can in fact be reduced to very, very basic, simple structures. So I just wanted to sort of get your read on intelligent design. Like, what do you think about it as a scientist, as someone working in a very complex biological structure that's difficult for many people to understand? How do you feel about intelligent design in this debate about how it stacks up against evolution by natural selection?
There is no endpoint in mind for evolution. It's really just a series of adaptations. It's not, a designer may have an endpoint in mind, I imagine. And perhaps your artists and designers in the audience they are the clock makers in intelligent design. They're the ones that have the vision, they have the input in mind. I think that random selective forces and time, and I think it's an open-ended, I think evolution is an open-ended process. And if you think about intelligent design, it has this impression of being a predetermined set of rules that's coming from where. As a scientist, I don't subscribe to the idea of intelligent design.
Yeah.
I could see how, it would be appealing to have some, some designer, some grand designer controlling.
Yeah.
There's revolution.
It definitely, I think it's interesting because it speaks to like a basic need that we have for meaning in our world and some kind of purpose. And that's, completely understandable. I am not a proponent of intelligent design either. As a designer, in fact, I mean, it's an interesting thing to think about. And I think you kind of mentioned it. One of the chief criticisms of evolution by natural selection, going back to like Darwin and the origin of the species, is that beautiful and really perfect structures would appear beautiful and perfect to us, right? How could they be made without any knowledge of how they were made or how they work? Because that's actually completely antithetical to the design process, right? Part of designing something is understanding at a very fundamental level how things work and then growing it growing the pattern from there, right? There's this philosopher, Daniel C. Dennett, and he writes about consciousness and the theory of mind. I know you're familiar with him, but he actually provides a different view, and he makes a really interesting claim, and I'm quoting him here. He says that all the design in the universe can be explained as the product of a process that is ultimately bereft of intelligence. In other words, an algorithmic process that weds randomness and selection to produce a unique, branching, recursively revised structure that bears as its fruit all of the intelligence that exists. In other words, evolution it iterates over many prototypes. It's very wasteful, like you said, a lot of time, a lot of creatures dying. You know, a lot of organisms didn't pass the test, and it passively tests all these prototypes in a massive environmental system. So in a way, like one of Dennett's claims is that evolution, though it's completely passive and rudderless and wasteful and has no engineer behind it, is still a kind of passive design process. It could still be called a design process in and of itself. Would you agree with that? Like, how do you feel about that claim?
It's interesting.
Yeah.
I would say that the rudder in evolution are the genetic material and the cellular material that within this framework,
right,
life exists. So it's not chaotic or random. It's structured based on the building blocks of life, and there are rules, the physical rules of the universe that guide evolutionary processes. So it's not in a vacuum either. I like Daniel Dennett. I read him when I explained consciousness. It sounds like he's in support of evolution as
absolutely
intrinsic intelligence and sort of with optimal output as sort of an emergent process.
Yeah, that's exactly what it is. It's an emergent process, which to us, we see the results of the emergence, right? So it's hard to tease out the fundamental processes that are at the root of all that. That is like the curse of emergence, right? It's hard to really, it's hard or sometimes impossible to tell, not impossible, very difficult to tell where it all emerged from. Dennett is completely a supporter of evolution by natural selection. And in fact, he relates that to why consciousness even exists. And I think his criticism of intelligent design is that it's not testable, right? And it doesn't provide any predictions, which is like how science works. Like unless you have something that can be testable and provide some sort of a set of predictions, and it's not really a theory, it's not even a hypothesis, it's really just a belief, which in some ways doesn't really have much place in the lab. But I think his like real claim is that the thing that is most valuable for me is that a completely blind and wasteful design process that is evolution, right, over millions of years led eventually to intelligence, right? That's what he works in consciousness and the theory of mind. And he's essentially linking evolution to how we got to where we are, how our brains evolved to where they are, and how intelligence evolved. And that eventually obviously evolved to designers, right? Me, right? To people like me that design things with intention. So do you think, if that's the case, do you think there's something primordial, and this is a big question, but is there something primordial about the instinct and the prerogative to design that we perhaps owe to this process of natural selection?
Well, yeah. I typically teach the students in the introductory neuroscience that the brain is a creative problem-solving computational system. So yeah, I mean, we evolved to solve problems, and I believe designing is a form of problem solving. We have problem solving as a survival mechanism, and mapping out reality and designing reality is something we've done for as long as our species has existed. We've modified the environment, and so you could call design certainly an emergent property of the human mind. We're not the only species that designs, but you have something other species that design nests, or other or in terms of reproductive success. There's bowerbirds, I believe, build
Right.
rainbow-colored nests to attract females. There's also
Yeah.
songbirds that diversify their song. I mean, these are other ways we see nature
Yeah.
using
Yeah.
design for Darwinian success. I don't know if the art students or you're using design as a mechanism for Darwinian success, but it's
Yeah.
it's in the context. You're in the context of life, so
I don't know how successful we would be, but
you're a professor. So
you never know. Another thing you mentioned, which was it was interesting to me, was that as the eye, and this is throughout the animal kingdom, throughout the life, as the eye evolved, I saw like a trend towards more detail, more resolution of detail. And I know that there are species in the animal kingdom that have higher resolutions than us, than humans, like birds of prey, for example, right? I assume have much higher because they need to. They have an evolutionary prerogative to see things at a distance and pattern recognition from a distance. But do you think that one of the things you mentioned was that the was it the phobia that allows us to like recognize details in faces, right?
Yeah.
Do you think, or I guess could you comment on the design of our eyes and how that sort of level of detail changed us as a species? Made us, for example, in the case of seeing faces, that's an important thing for us, a garrulous species, a social species, and just a social network to evolve. Could you talk about maybe other ways that the structure of the eye and our eyes has changed us from an evolutionary standpoint?
Yeah, that's a good question. I mean, it massively, obviously, it's influenced human culture around over known history, from cave paintings to degree programs in the fine arts to advertising. Obviously, our high acuity for color and detail is used, obviously, for the purposes of arts. And I can see many ways that the structure of the retina has shaped culture, or at least provided. We also, not just the retina structure though, David, it's obviously we have the brain mass in the visual cortex to computationally analyze more information than any other species. We have a large, what's called an encephalization quotient. We have a very dense, convoluted. If you were to take out the cortex of the brain and unfold it, it would be like a 3 foot by 3 foot sheet of neurons that are really densely compressed. So we have it's the architecture of the retina, but then the architecture of the visual cortex that gives us high visual acuity and high visual processing and analysis. I'm not sure if I answered your question. That was a big question that you asked.
No, I think it answers the question. I mean, it's culture, right? I mean, it led to a culture, and that, I mean that like capital C. It's like everything, like language, right? Visual communication, even just social interactions and social cohesion was made possible in some ways because of our eyes. Like they're incredibly important. They evolved from what you're telling us to increase our success as a species in terms of reproduction, right? But that means a lot of different things to a lot of different people. Like it means on a to an anthropologist that means hunting, perhaps, right? Or better hunters because they could see pattern, they could distinguish patterns better and movement better. But it also means, like, to a linguist, right, it means being able to read and write and understand, and that's in conjunction with our brains being able to manipulate symbols and think abstractly, obviously. But none of that matters if we don't have the eyes that to process and help us see this information. This leads me to another question: is that humans have been incorporating technologies into our lives for hundreds of thousands of years, if not longer. And I, for example, I consider language to be a technology, right? It's a it's a tool we use to communicate and to, it's a skill, right? This is becoming, like, just the other day, Facebook, for example, announced that they were rebranding themselves as meta, right? Which is they're trying to conquer the metaverse, which is a virtual reality space where we communicate in a virtual realm. We have virtual selves, etc. And it requires a lot of technology to make this work. It requires cloud computing and servers, and but it requires augmented reality, mixed reality interventions essentially into our eyes and our brains and our daily lives. So do you think that in the same way that, maybe like prokaryotes, I'm spitballing here, but they absorbed basic nuclei, became eukaryotes, and then basic cells absorbed mitochondria and made them part of their this endobiosis idea that some larger entities absorb smaller entities and they become one synthetic organism. Do you think that eventually this is what's going to happen, or it maybe already is happening with these technologies in our daily life, that we'll eventually absorb them and we'll become indistinguishable from them?
I think it's already happening as far as I can tell.
Mm-hmm.
Future's happening. Technology, you mean information technology and artificial intelligence, and
Mm-hmm.
absolutely, yeah. AI is big, and neural interfaces are, I believe a couple of companies are working on neural interfaces. We're working on retinal prosthetics. I think the future is happening now, and an incorporation of, I think we will adapt to incorporate the new technologies that you're describing, and it will enrich our lives. I assume that I would think that the human brain is benefiting from the increase in information available in this technologic era, and you wouldn't want to go backwards.
Mm.
So is that answering the question?
I suppose it's a little bit more positive than I am. It's like more glass half full, that in more information is better, right? Like more detail and more ways of looking at things is on the surface a better thing.
And then the mind can function as a selective filter. You would rather have the light than be in the dark.
Yeah, so I suppose that is
Hmm.
that's a very good answer then. Yeah, I think that does answer the question in a very elegant way.
Thanks.
One other question, this is about art, because I'm asking you this because you have a background in the arts. You were a sculptor, you still are a sculptor. I mean, you never stopped being an artist, right? But you might stop practicing or do other things, but it's always there, right? As a communication technology, art's possibly older than language, the spoken or written language, right? We don't know. I don't know. I'm not an anthropologist. I have no idea. But what is it about our eyes? And you kind of suggested this, or I don't know, you kind of went into this a little bit in your presentation, but what is it about our eyes, and in conjunction with our visual cortices, that invites visual play and communication? Is there something there that it was an inevitability that we would become visual species, visually driven, and artists?
It's a big question, Steve.
Mm-hmm. I'm hitting you with the big ones.
Good stuff, though. Good stuff. If you ever have had the experience of appreciating beauty, perhaps you can see that we have some hardwired, genetically encoded, evolved appreciation for beauty that gives us a sense of reward, whether it's beauty in our own species or, beauty in a Jackson Pollock. So I think there's some appreciation for symmetry.
Mm-hmm.
And I guess the definition of beauty is this sort of malleable, but I think everyone would agree that it is a part of our consciousness. Visual consciousness does have a component for the acknowledgement of beauty. I think Dennett might agree with that.
Yeah, I think so. Yeah, it's just weird to me, because you look at other animals, even other primates, right? Like the closest related primate, like the chimpanzee, they have nothing approaching what we would consider to be aesthetics or art of any kind. They don't really have, I mean, they can communicate, other primates can communicate. So they do understand abstraction, but in terms of like externalized expression, they have nothing like that. Unless I'm wrong, I mean, unless I'm sorely mistaken here. And I just find that very bizarre that we're like some kind of aberration. And I'm just wondering, does it have anything to do with our eyes, in our visual cortices, obviously, and the extent of our brain that you've already made clear. They have eyes too, you know, the chimpanzees, gorillas. Are their eyes that different than ours from your experience?
They're quite similar, but I would say that, you know, let's not underestimate the potential for a chimpanzee to appreciate beauty in another chimpanzee. I'm sure that's happening.
Actually, yeah, there's a comment here. There's a comment here that you are wrong. Chimps will dress up if provided with materials. So, and then there's also, whales have songs, which is true. And I know that whale songs, they're very complex and they repeat over a period, like entire decades. So that is true, yeah. There's a couple comments here I just wanted to read. We have a little bit of time here. Let's see. Let's see here, yeah. The real difference, I'm just reading something here. The real difference comes when you take the time to see. When you truly see, you allow your imagination to see more than what is before you. It brings to mind something said by Dante that fact is the enemy of truth. I believe this philosophical sense of looking at one thing and seeing another is the essence of mental and visual exploration. In the end, I hope my students will go through life seeing wonderful things. This is, of course, Sara Little Turnbull. She's the namesake for this initiative. And that's an interesting perspective here. And lastly, when can you tell us about what you are doing in terms of in your lab, like what you're doing to develop for the human eye and to assist it and perhaps improve it?
Sure. Yeah, we can go on back to the science. It's been lots of fun though so far. So I give you a video here, just to briefly. We have a little time. And this is the engineering of a retina in a dish. At this point, it's quite miraculous that about 2 decades ago, after pushing stem cells for many decades to try to become neurons individually, a group of scientists took skin cells, incorporated 4 pluripotent genes. These genes are pluripotent, meaning stem cells are pluripotent. They can become any cell in the body. If you take an adult skin cell, it's already a skin cell, but you can inject those stem cell genes into it, and it becomes what's called an induced pluripotent stem cell. This was a very big breakthrough not very long ago. And more amazing than that, when I was doing my research up in Boston, trying to engineer retina tissue from polymers, which is on the next slide if we have time, a group came out with this paper that showed that what's happening up here in the top right corner is that you can take induced pluripotent stem cells, put them into a petri dish, and trick them into believing that they're developing into a retina in the dish by adding different growth factors. And these very elegant and completely stratified retina develop spontaneously through self-assembly in the dish. Now, I give this to the group to show you, we have self-assembly in limpets and self-assembly in the evolving eye. And then this has been tapped into recently in what I would call cellular engineering to build a retina, which it sort of happens like a domino. They're pushing over the first few dominoes by adding growth factors to pluripotent stem cells. Then this diagram shows that they're going from stem cells to embryoid bodies to developing a neuron identity, then to becoming these fascinating retinal cups. So we do some work on this. I work on looking at the signaling that's involved, but I really just want to present this to the group to show you one of the most cutting edge technologies in retinal engineering that wasn't conceivable 20 years ago. And this is video microscopy. If you're visual people out there and you're wondering what is that green blob glowing in a dish, those are jellyfish genes that have been added to induced pluripotent stem cells. And then this is a time-lapse movie being made over probably several weeks, sequentially taking steps like stop animation and then stringing them back together. And then you have this three-dimensional picture of the growing retina in a dish. So I do a little bit of work on this stuff, and I'm not sure if I have time for tissue engineering, but what I did when I was a postdoc before I started my lab was some of this tissue engineering. This might appeal to the designers in the audience, David. You want to develop smart materials, especially when you're trying to develop a neural tissue from stem cells. And one of the major strategies is to make the polymer or make the biomaterial biomimetic of the retinal tissue in vivo. You know, what kind of growth factors are present? What is the substrate like? Is it hard or soft? You know, what kind of proteins are present? What kind of growth factors? So when you make a biomimetic polymer or a biomimetic material to engineer nerve tissue, you take everything you can from developmental biology and from anything that can be extracted from the mature nervous system, and you try to incorporate it into your biomaterial. And that's shown here when it says engineered materials. At the top three circles say natural and synthetic, biochemical modifications, and mechanical properties. You know, if you want to make a neuron, then grow it on very soft biomaterial. If you want to make a bone, then take a stem cell and put it on a very hard biomaterial. And the cells have enough intelligence to sense that they're on a soft tissue or a hard tissue, and they activate gene expression programs to adapt to that environment. There's some real smart cellular intelligence in stem cells. And then, you know, you can move around the circle here, and you have emerging technologies. You have some other properties like electrical stimulations we've done. You know, you can add different growth factors and the timing of the stem cells exposure. In the end, when you start to look at what is the end output of this project is, you know, you have stem cells that you want to generate into nerve cells probably for cell replacement or for drug testing. And so you want to find a high throughput and reliable way to convince the stem cells that they're becoming neurons and to complete their process of becoming neurons. Any slight deviation from what they're expecting can lead them to die, to cell suicide. But in the end, you know, right now we have some very powerful tools. We have tissue engineering, and we have the 3D retinal organoids. And let me go to the last slide I have here, because it's got a nice video. When I was just starting my lab, I was working on this, and this was in collaboration with someone named Robert Langer at MIT. He's quite well known in the biomaterials field. And you can make something called a polyglycerol sepicate, PGS at the top, and it degrades by hydrolysis, just by water, slowly over the surface. And that's good if you want to put something into the nervous system. You want it to slowly dissolve without killing the surrounding neurons. So we made these hard molds at the bottom, A and B, show you the hard molds. You coat the hard mold, the plastic mold, with sugar at the bottom, and then you put your layer of polyglycerol sepicate on the top, and then you peel it off. That's the green scaffold at the right. And then this is a scanning electron micrograph showing you what one of the holes looks like, which was really fascinating to me coming from an artistic background, to see the three-dimensional, tangible structure of these biomaterials. And if you are an artist and considering science, I think biomaterials work is really intuitive for us, or for you. And then you can see these four holes. These are where stem cells have crawled into there. You culture the stem cells or the neurons on top of your polymer, and then they express these different colored markers that say they're becoming neurons. And then I made this movie myself when I was probably 35. This is one of the polyglycerol sepicate coated scaffolds covered in green neurons that are then going to be injected into the mouse eye. And this diagram here shows you how you would inject a polymer coated with new neurons underneath the human retina. And that was really compelling and engaging work, and very hands-on as a scientist. It's very visual, and I really enjoyed working with the bioengineers and the biomaterial scientists, because I would just come in with my imagination, which you could do also, Dave. And I would say, I want a scaffold with holes in it, or I want a cage to put my neurons in so that they don't crawl away. And they would, in a week or two, they would have it. They're so skilled at developing these micropolymers. And that's this slide, and then I have a conclusion slide. We could stop here, or it's just, so you have, if we go back in this brief conversation, we have early eye spots evolving through exaptation, which is a novel process of where natural selection uses parts unexpectedly. And this was to provide phototaxis in single cell eukaryotes. And then we talk about the evolution of the eye from the limpet to the octopus to the primate eye. And you'd say natural selection favored gene combinations to improve retinal function and preserve the life of the species. And the life of each species is different depending on its environment, where the limpet eye still serves it perfectly for its survival. The end goal is not that all species should have a complex eye, but that their eyes should serve to preserve their life in their given environment. And here we have the mammalian eye has resolution, direction detection, light and dark sensitivity due to the structure of the pupil, lens, cone, and rod and photoreceptor arrays. And what I just showed you is that retinal architecture can be recapitulated using induced pluripotent stem cells and self-assembly retinal organoids. And if you wanted to get into biomaterials, you can drive neural differentiation and improve delivery of nerve cells to the eye using smart polymers. Those are my conclusions.
Excellent. I have a couple questions from the audience.
Yeah.
How does your work inform your art? Your research in the eye inform your sculpture work, which I don't know how much you engage in at this point, but
my current work in neuroscience and
yeah,
I do sketches now, and but there's not a direct correlation between my science and my art. I don't know if there should be a direct correlation between science and art. I think there's some beautiful areas of art that don't need to be quantified or literal. But we can talk more about that. It's a big question,
Oh yeah, and also your medium. What when you said you were working in sculpture, but what were some of the media that you worked with?
Oh, I have an image here.
Sure.
I was apprenticing for a bronze sculptor, a neoclassical bronze sculptor, as I mentioned with David, named John Saunders. He's the director of public sculpture in New York City now. So I was working in bronze. I started in clay, and then I worked in mixed material. But I did learn the lost wax method, which is an ancient method of making a clay armature, covering in wax, covering that in porcelain or ceramic, and then melting the wax, and you have a ceramic shell that you can pour bronze into, and you roll it so it becomes a hollow bronze sculpture, and then you crack the ceramics, and you have this is how old bronzes were made, and this is what I was doing when I was before I went back to graduate school.
I think this is interesting because it parallels in some ways a couple slides before where you were talking about the materials, and they would make materials by additive and then subtractive processes to get the mat where you could then grow stem cells into larger structures. Like it's very, very similar. It's just that this is at a macro level versus a microscopic level. So I think that if there is a connection, it's there. It's possibly there. I think that's really fascinating. Yeah. And lastly, I just had one other question for our students, for the purpose of our students, that you have a lot of in your presentation and in your the different articles, the reports that you've published, a lot of illustrations of biological structures. There's a lot of information design in terms of charts and graphs and et cetera. What kind of demand is there, and why is that even important? I mean, aren't scientists just really interested in data? Like, why do we need to see beautiful pictures of these things?
Well, that's a great question. And I'm open to art students emailing me for collaborations. We need visual scientists to visualize the paradigms we're describing. Oftentimes we're describing paradigms that haven't been previously described. And so one software we use is called Maya. And you're probably familiar with it as art students, but molecular animations are valuable because it gives you an immediate understanding of the intricate molecular processes you're trying to describe. And also scientific illustrations are important. Many journals, if you're going to publish your scientific work, will ask you for a visual abstract. And that's just an intuitive, artistic rendering of the story you're telling as an artist. So it helps with our comprehension that we work with artists, and otherwise it gets quite complicated. And an image, is quite helpful in that way.
Yeah. Cool. Yeah. Well, this has been really really interesting. I particularly am interested at the bottom left here. You've really linked artistic mechanisms, artistic technologies, sculpture technology to the things that you were talking about previously. I find that fascinating. And I think perfectly answers this question, right? Oh, and lastly, just one other question that's popping in. Did you create the images in the presentation for today? I saw some image credits in some of them.
Yeah.
Do you do the illustrations in your reports, or do you commission those, or like how do you arrive at those?
Both. Sometimes I make them and sometimes I commission them, or if I use images from publications, I'll reference them. Whatever I wanted to communicate today, I found the most precise illustrations I could to communicate that. A couple of images of mine where I was describing my publications.
Yeah.
So
cool.
Whatever. Yeah.
That's wonderful. Well, thank you so much. We're just about out of time. Thank you for visiting with us today. We really appreciate it. And so just everybody who's in attendance, our very last lecture in this series is going to be next week, where we're going to be finishing out this discussion of the eye. I hope you'll all join us. Thank you, Professor Redenti. I hope you have a good rest of your day. And thanks for everybody that came and attended today.
Thanks, Dave.
Great. Thank you so much. All righty. Everyone have a wonderful day.